everybody. This is Dewey McCafferty. I'm a professor in the chemistry department at Duke University, and I wanted to welcome you to our research lab. We focus on chemical biology here at Duke, and our interests are in a lot of different areas that lie at the interfaces of chemistry, molecular medicine, and biochemistry. And so, in the next couple of minutes, we'll take you through a tour of the lab, let you meet some of the folks, and learn about some of the exciting science that they have going on.
The McCafferty Lab offers training in a variety of chemical disciplines, including chemical biology, medicinal chemistry, organic synthesis, biochemistry, and enzymology. All of our projects are highly interdisciplinary, and students have the opportunity to gain a wide variety of technical skills. Currently, we have 3 major projects surrounding Parkinson's disease, chlamydial pathogenesis, and lipid tube binding natural products.
In recent years, we've become interested in the chemical biology of Parkinson's disease, a prevalent neurodegenerative motor disorder with complex and heterogeneous causes. Our interest in Parkinson's began when we repurposed a library of small molecules that we found were able to stimulate distinct neuronal signaling pathways and showed efficacy in mouse models of Parkinson's. While there's still work to be done in this area, we've more recently begun exploring the underlying causes of Parkinson's by studying the mechanism and targets of small molecules that address associated cellular defects. This work has taken a truly interdisciplinary approach using a wide array of techniques, and we've been fortunate to have funding for both the research and the students working on this project from a variety of sources.
Another major research area in the lab is Chlamydia trachomatis, the pathogen responsible for the most common bacterial sexually transmitted infection worldwide. Our work is focused on investigating enzymes involved in the bacterium's complex, obligate intracellular life cycle to identify and characterize novel anti-chlamydial targets. A foundational project on the protease CPAF, which is exported into the host cytosol to promote evasion of the host immune response, involved elucidation of the catalytic mechanism of CPAF and identification of selective small molecule inhibitors. More recently, we have begun investigating the biosynthesis of menaquinone, which is an electron carrier required for ATP production. Our current efforts are geared towards cell-based assays with chlamydia infection models, functional validation of enzymes through mechanistic enzymology, and the design and synthesis of small molecule inhibitors. Collectively, the chlamydia projects encompass a wide variety of techniques and skills to improve our understanding of this public health threat.
The antibiotic project within this lab is fueled with this desire to understand how compounds referred to as natural products are able to inhibit bacterial growth via inhibition of the late-stage cell wall intermediate, lipid 2. We use a variety of techniques in order to elucidate how the structures of these antibiotics relate to bioactivity. This includes using computational tools to investigate biosynthetic gene clusters within novel bacteria to produce conjugators of the natural products. Further, we use enzymatic techniques in order to interrogate how these natural products are produced and utilize these tools to create second-generation antibiotics that have improved pharmaceutical properties. Within this project, we use a wide variety of biochemical approaches, such as structural elucidation, enzymatic assays, and bioanalytical techniques.
I'm Brianne. I'm a 5th year in the lab from Pittsburgh, Pennsylvania on the chlamydia project, and outside of the lab I like to play the piano and go to Duke basketball games.
I'm Katie Hatstadt. I'm a 5th year from Parker, Colorado, and I obviously love chemistry, but I also love to play soccer and to go running and to hang out with my dog.
I'm Lydia. I'm a 4th year in the McCafferty lab. I work on the antibiotic project, and I'm from LaSalle, Illinois. Outside of lab, I like to take part in intramural sports here at Duke.
I'm Taylor. I'm from Clayton, North Carolina, and I enjoy coaching cheerleading at a local high school.
Hi, I'm Natalie Schulte. I'm a 1st year in the lab. I'm originally from Delaware, Ohio, and a fun fact about me is I love to sew.
Hello, my name is Hunter DeHaan. I'm originally from DeMont, Indiana, and an interesting fact about myself is that I can play 3 instruments.
Hey, I'm Mike. I'm working with Katie on the Parkinson's project. I'm originally from Holly Springs, North Carolina, and I'm finishing up my senior year right now. Outside of lab, I enjoy listening to music and playing the guitar.
My name is Charlotte, and I'm an undergraduate senior. And outside of the lab, I like to go to Duke basketball games and run on the trails here in Durham.
My name is Michaela Reinhardt. I'm a senior at Duke,
from Georgetown, Kentucky. I love to play spikeball and run outside of lab, and fun fact, I have a wiener dog named Tater.
Hi everyone, my name is Jeffrey. I'm an undergraduate from Shanghai, China, working in the lab, and in my free time I enjoy cooking.
Why I chose Duke, besides the great faculty and research here, was the grad students themselves. Each one of them that I met over the recruitment weekend provided a great sense of community, and they were all such wonderful people that I knew this would be the place I would spend my next 5 years getting a PhD.
I decided to come to Duke because when I visited, I felt like there was a true sense of community between the students and faculty. With such a small program, you get to know a lot of the people, and then you also not only get to learn what the chemistry they're doing, but you get to know who they are outside of lab, which I really appreciated.